Hello! Today we are studying microbial growth, and this is part 2. If you have not seen part 1, how to study microbial growth, then you can check that out on my Instagram page. Yesterday we studied, what did we study? Temperature, microbial growth, and the effects of temperature on their growth. Today we are studying the pH, the effects of pH on microbial growth. So, hello, thanks for joining me. Feel free to comment and share and give hearts if you find this very helpful, informative, and you enjoy it. So we are going to get started. I'm going to flip this around and adjust it. Oh, I keep forgetting to introduce myself. My name is Rochelle, and I am your microbiology study strategist, where I position you to understand, retain, and remember microbiology. And that's what we are going to do today. All right, let me flip this around. Adjust. That should be good. If this falls, my bad. So it is the microbial growth, and we are talking about the pH and the effects of acidity and alkalinity. So we are going to start with the acid. Well, let's explain. This is the pH scale, 1 to 14, 1 being acidic. I should change the color to red because I like color coding. And 7 range being neutral. And 14, well, what color? I use that color. 14 being alkalinity, being very basic. So that is the scale. Now we are going to talk about the microbes that are within each group, starting with the acid. So there are some microbes, they are called grouped acidophiles. And some of them are, I'll name a couple: Theobacillus, acidophilus. Another one, let's see, a famous one that let me adjust this. So a famous one that we would know, and we find this in yogurt, and it's lactobacillus. Acidophilus. Is it 2? Phyllis. U.S. Okay. Boom. Boom. And you see that in yogurt. Hey, thanks for joining. You see that in yogurt. You also can find this bacteria in your mouth, your intestine. Oops. Back. And in the vagina. All right, let me move this up some. There. So that's where you can find this particular acidophile. You can also find them in the deep sea hydrothermal vents, sewage pipes, sulfurous waters, and the Yellowstone National Park. Some examples of where you can find acidophiles. Next, we are going to talk about, and let me scroll this down so you can see here. You can see this. Hi, thanks for joining. Hello, thanks for joining. So we are next going to talk about neutral. Neutral was called neutrophiles. So neutrophiles. And I'll name some of the bacteria. So we have E. coli, Salmonella species. What else? Staph. You can also find staph. Leucococci. And then the environments that you can find. Of course, you can find it in humans, especially E. coli. You can find E. coli in your intestines. You can also find Salmonella out in nature, in soil. So that's where you can find in soil, nature. That's where you can find these particular microbes, neutrophiles. Next, we are going, one more thing that I wanted to add is most bacteria, their growth is between 6.5 and 7.5. So that is important to know. Let me write it right here. So 6.5 to 7.5, that's where they grow the best. And we are going next to alkalinity. Now alkalinity, go to blue, no, this one. All right, with alkalinity, the microbes that we see here are Vibrio, and this is a very famous one, a well-known one that most people know because this particular microbe causes cholera. And you guys should be familiar with cholera. If not, you can get cholera from contaminated water, a contaminated water source. And that contaminant is feces. So if there is feces in the water, your water is contaminated and people can get sick. Another microbe, it's an archaean, and it's called Natrona bacterium. That's another one, and you can find this microbe in the soda lakes of Africa Rift Valley. So that's where you can find this particular microbe, and these are under the group alkaline. So alkaline phials. All right, so we have acidophiles and neutrophiles that we already covered. We covered the microbes and the environments that you can find them in. So here, contaminated water. Oh, you can't even see. There you go. Contaminated water. So with this one, it's contaminated water. And usually you'll see it's feces. All right, what else do I want to cover? Oh, another one in the neutrophil family, relating it to, let me scroll down, to a real life situation. I know people heard of ulcers, stomach ulcers, or it is formally known as peptic ulcers. So peptic ulcers or stomach ulcers. You see, most people would get this, and back in the day, doctors would say, stop eating spicy food, just regulate what you eat. They didn't realize what was actually causing stomach ulcers, and it was a microbe that was causing stomach ulcers, and that microbe is called Helicobacter. Am I spelling it? Yep, Helicobacter pylori. That was the microbe that is the microbe that's causing stomach ulcers. Now, what it does is it creates, it is a neutral. So we know that in our stomach is very acidic. So our stomach is on this side. It's very acidic because it does have HCl, hydrochloric acid. We have that in our stomach, so we know that it's very acidic. Now, and they thought that Helicobacters were acidophiles, but they're not. They are neutrophiles. So how could a neutrophile bacteria live in your stomach? Or actually survive in the stomach? Well, they can survive because they create an enzyme called urease that breaks down urea. So this is my little breakdown symbol that breaks down urea and provides an environment around itself that is neutral. That is neutral, and it can survive in the stomach. Now, how to cure or actually treat, a treatment for ulcers is antibiotics. Now, how could scientists create antibiotics? Well, in order to create antibiotics, they would have to know what bacteria is causing the disease, the illness, the sickness. And when they know what the particular microbe is, they would have to know its structure. And once they know the structure, they can create drugs, antimicrobial drugs, to attack the structure of the microbe. And when the structure of the microbe is attacked, then the cell will lyse, or the function will decrease, or it would actually stop. Would actually stop. And if the function stops or decreases, then the growth decreases or stops. And you're healthy again. Then you're healthy again. So going back to chapter 4, knowing the structure of the cells, and knowing what feeds into the cell, knowing all about the cell is very important. So building that foundation from the beginning of the chapter till now is very important, because this is how they create antibiotics and create these antimicrobial drugs to treat illnesses. So that is a real life situation, real life example that you can take with you. Another thing that I want to talk about is, let me change the color to orange, is that not only bacteria has a pH level where they can thrive in, we also see mold and yeast surviving at a pH level of 5 to 6. So between that range, they are able to survive in that, in that range. Now, in a lab sense, a lab example of how microbes are grown, let's do this color because it's pretty. Here. So we have our lab, and we want to grow our bacteria. So lab growth, we want to grow our bacteria. So we have our bacteria, and our bacteria produce acid. And when they produce acid, then they aren't able to grow as well as we want. Because remember, I said that let's scroll back up, most bacteria grow in this area, 6.5 to 7.5. If their environment becomes acidic here, then they aren't able to grow. So how are we as scientists supposed to neutralize and allow our bacteria to grow? Well, in the media, you are supposed to. So there will be, the growth would stop. Growth. So what we do in the media, when we culture it, we add buffers. So we're adding buffers. I guess let's see. All right. We add buffers, and these buffers are peptones, amino acids, and phosphate salts. Now, phosphate, this one is a good one because it has phosphorus in it, and phosphorus is one of the essential nutrients that bacteria would need to grow. So yeah, this one is a good one to use as a buffer, and it will neutralize the acid and then increase growth. So that is all the information that I would like to share. So then, how do you study this? How are you going to retain it? Well, I'm so fond of Micromind Mapping. So what I would do is, let's see, I would have three Micromind maps, where one, it's all about acid. Then you'll have your bacteria. I can't even see it. So you'll have, let's see if I can move it. Yes, I can. All right, so you can have your bacteria. You'll have the environment. What else? What else can you add? You have the acid, the level, the pH level, and any other information that you want to add, you can add. Well, those are the main three things. And then with bacteria, it can stem to specific ones and different environments that you might come across, and maybe like the optimal pH level that you can put as well. So the sky is the limit. So that's how I would study this part. And then again, you would go to, and plus I like color coding, so I will put this all in red for acid, and then for neutrophils, I'll do the same thing. Bacteria, environment, and pH level. And then I would practice this over and over and over, drawing it, or I did a video on making flashcards for this, making it into a micro puzzle. So you can do that to help you retain it. And to help you remember it, I have questions that I posed yesterday that you can answer. So yeah, make this a little bit bigger. There. Questions that you can answer. Question number 1, what type of microbes are able to tolerate acidic environments? I covered that today. What is the optimal temperature of pathogenic bacteria? I covered that yesterday. And then which organism can live in a hypertonic environment? And I will cover that tomorrow, but if you know the answer, feel free to put it in the comments. And that's how you can study microbiology, how you can study microbial growth. Knowing the information, Micromind mapping it, and just committing it to memory, and then answering questions to test your knowledge. And that is it. Hey, I'm glad that you joined. I was thinking about your answer that you posed. I think it was for 1. You said extreme files, and you are right, because extremophiles are like a group, and in that group are haliophiles and thermophiles, and they can grow and survive in acidic environments. I was thinking about extreme thermophiles and extreme haliophiles, but I want this answer for number 1 to be extremely specific to what I taught and what we studied yesterday. So you are right. Just I was looking for a specific answer. And I think that's all that I have for you guys today. I thank you for joining my live. And let me turn this back so you can hey. Yeah, I was looking for a specific answer to that question, but in a broader sense, yeah, the answer is correct. But yeah, if you found that this lesson was very helpful for you, feel free to click the link in my bio, because this is what I do. I teach, I study with you, and I test your knowledge to make sure that you actually understand what you are studying, what you are reading. Click the link in my bio, because that's what I do in my Sunday sessions. I tutor, teach, study, and test your knowledge. Oh. Yes. What else I want to say? There's something else. If you don't need help understanding, but your study structure is like disorganized and you need help putting it together, you need help trying to study microbiology, I have a step-by-step how do I? No, it's okay. We're all learning. I don't know everything about microbiology, but it's an ever learning process, a discovery. I mean, that's what science is all about. It's about studying and exploring the information and testing that information. Yeah, and researching that information. And so if you're having trouble structuring your study time, I have a a custom study plan for you that will actually not one, but actually 4 weeks worth of custom study plans for you so that you can go into each study time knowing how to study, what to study, and you're not confused. You're not somewhere confused, lost. It's all ready for you. And how you can get that is by clicking that book now button on my page, on my Instagram page, where you can book your time, and then we can talk about what your schedule is looking like, and I can create 4 weeks of custom study plans for you so that you can go into your study time knowing what to do and that you're not lost. Thanks for the hearts. So click the link in my bio if you want to join my tutoring sessions. If you need a custom study plan in place for your study time, then you can hit that book now button, and I can do that for you. Any questions, you can put it in the comments or you can DM me, and I will talk to you tomorrow for part 3, the last video of this series. All right, talk to you guys later. Bye.